Welcome to our channel. Today we're diving into the fascinating world of blood histology. Blood is a unique tissue composed of various specialized cells circulating in plasma. We'll explore the different types of blood cells, their functions, and how we study them using techniques like blood smears. Let's get started. Blood is made up of three main components: plasma, red blood cells, and white blood cells. Plasma makes up about 55% of blood volume and consists of 92% water, 7% proteins, and 1% other solutes. The remaining 45% of blood volume is composed of red blood cells, white blood cells, and platelets. Red blood cells, or erythrocytes, are responsible for transporting oxygen from the lungs to the tissues and returning carbon dioxide from the tissues to the lungs. Erythrocytes are biconcave discs that lack nuclei and organelles, allowing for maximum space to carry hemoglobin. Their shape provides a large surface area for gas exchange. White blood cells, or leukocytes, are the body's defense against infection. They are classified into two main groups: granulocytes and agranulocytes. Granulocytes include neutrophils, eosinophils, and basophils, while agranulocytes include lymphocytes and monocytes. Granulocytes characterized by the presence of granules in their cytoplasm. Neutrophils are the most abundant type and crucial for fighting bacterial infection. Eosinophils are involved in combating parasitic infections and allergic reactions. Basophils release histamine during allergic responses. Agranulocytes lack visible granules in their cytoplasm. Lymphocytes, which include B cells and T cells, are central to the adaptive immune response. Monocytes are the largest white blood cells and differentiate into macrophages and dendritic cells to engulf pathogens and present antigens. Platelets, or thrombocytes, are small cell fragments crucial for blood clotting. They gather at the site of a blood vessel injury, adhere to the vessel wall, and work with clotting factors to form a blood clot, preventing excessive bleeding. To analyze blood cells, a blood smear or blood film is prepared. A drop of blood is placed on a slide and spread thinly across the surface. The smear is then stained using right stain, which differentiates the cells and highlights their structures. When examining a blood smear, the best area to analyze is near the tail, where the cells are evenly spaced. Erythrocytes appear bright pink due to hemoglobin, while leukocytes and platelets can be differentiated by their unique staining properties and morphology. In summary, blood histology is the study of blood cells, each with unique structures and functions. From oxygen-carrying erythrocytes to infection-fighting leukocytes and clot-forming platelets, understanding these cells is crucial for diagnosing and treating various medical conditions. Thank you for watching. If you enjoyed this video and found it informative, please like, share, and subscribe for more educational content. Have any questions or topics you'd like us to cover? Drop a comment below. Stay curious and keep learning.